I'd like to get started first by welcoming all of our participants today and also welcoming our speakers to the first session of the Where Are They Now HQP Alumni Career Series. My name is Megan Mahoney, and I'm the Director of Scientific Affairs and Training Programs at BioCanRx, Canada's immunotherapy network. So before we jump into the moderated session with our alumni, I'd like to just provide a few housekeeping notes. The first being that this session will be recorded for the majority of the session. With that being said, we're going to stop the recording about 15 minutes prior to the end of the hour-long sessions to allow for participants to unmute themselves, ask questions, and potentially have a conversation with the panelists that we may wish to not have recorded. With that being said, if you have any questions throughout the entire session, please feel free to ask them. There are a couple of ways of doing this. So the first way is by asking a question in the Q&A box, and that allows other participants to view it. And you can ask that question anonymously, and other participants are able to upvote questions that are of interest to them. Secondly, if you wish, you can raise your hand and ask the question orally, and we welcome both. I think one of the keys of a successful session like this is interactivity, so we certainly promote that. And secondly, and building upon what I just described, for the majority of the session, you will be muted and you won't have video. So again, I just want to reiterate that if you do want to ask a question orally, you raise your hand and I'll unmute you. If you're having any technical issues, please write that in the chat box and we'll address those as well. So with that being said, I would like to, again, welcome our alumni. For those of you who may not know, this series was started first based on a Q&A question that was asked of these alumni that was in written form, and we can provide that link later on in the session. But effectively, we asked the various different alumni questions, and now we're checking in with them just to see where they currently are and to get a little bit more of an opportunity to speak with them. So without further ado, I'd like to introduce the first panelist, and that person is Piriya Yoganathan, who is currently the head of research and development at the Biotherapeutics Manufacturing Center at the Ottawa Hospital Research Institute. She completed her PhD at Queen's University in the Department of Pathology and Molecular Medicine with a specialization in cancer research, and she also holds an executive MBA from the Telfer School of Management at the University of Ottawa. And I should say that the screen does not present how I'm describing it. So we'll now jump to the furthest left. That's Laura Evgen, who is a scientist at the Genome Sciences Center at the BC Cancer Agency and is an assistant professor at the University of British Columbia in the Department of Medical Genetics. Laura obtained her PhD under the supervision of Dr. John Bell at the Ottawa Hospital Research Institute, where she studied oncolytic viruses. She then undertook a postdoctoral research fellowship at the Mayo Clinic in the lab of Dr. Richard Weil from Duke University to evaluate combination approaches using OVs and CARs. She was recruited to Vancouver in 2020, and her research portfolio includes the use of viroimmunotherapy and CAR-T for lymphoma and other solid tumors. And last but not least is Dr. Parvez Vora, who is the site head of Century Therapeutics Canada, an innovative biotechnology company that harnesses the power of adult stem cells to develop curative cell therapy in immuno-oncology. Before that, he co-founded Piriya Therapeutics along with Dr. Sheila Singh from McMaster University and Dr. Jason Moffat from the University of Toronto. Parvez earned his master's degree from the University of London, UK, and a doctoral degree from the University of Manitoba. He then trained at McMaster Stem Cell Center and Cancer Research Institute in Dr. Singh's lab as a postdoctoral fellow, and then subsequently as a program scientist. So with that, I'd like to welcome you all here today. I will stop sharing my screen. And we can dive into it. So on my screen, the order is Laura, followed by Piriya, and followed by Parvez. So the first question to all of you is really just to provide a little bit more of a bio, anything I may have missed or anything that you think was important to mention at the offset. So tell us a little bit about yourself. And so, yeah, thanks, Laura.
Yeah. Hi, so thanks for having me here. I'm happy to share my story, and hopefully that will be useful for other people to navigate their own path. So I'm particularly interested in immuno-oncology, and I did my PhD with John Bell in Ottawa. Like Megan mentioned, I studied the systemic delivery of oncolytic viruses. And from there, I got really interested in cell therapy, and I undertook a postdoc at Mayo Clinic with Richard. And there I learned to work with mouse CAR T, and I was just, really taken with the subject, and, decided that I wanted to set up my own research lab. And so I was recruited. I did my interview virtually. I was recruited to UBC. I never actually came here before. I never was able to come for my interview. So we just showed up, and I've been building my lab since November of 2020, and it's been, really exciting, and also filled with lots of perhaps anticipated and also many unanticipated challenges.
Thanks, Laura. And I suspect we'll ask about some of those later on in the session. So Piriya, I'll turn it over to you too.
So yeah, first off, thank you to BioCanRx, particularly Megan, for the invitation to participate in today's panel. A little bit about me. I've always been very passionate about science. I completed a BSc in cell biology and genetics at the University of British Columbia. And while at UBC, I had the opportunity to work on a couple different metabolic research-related projects in the labs of doctors Suzanne Klee and Jan Isis. And that research experience that I had at UBC was very foundational and inspiring for me to continue on in the field of research, and in particular was interested in extending into cancer research. So was drawn to the Queen's University's Collaborative Cancer Graduate Program. It has a transdisciplinary focus, so not only focuses on the basic science, but also the epidemiology as well as clinical trials. And so joined Dr. Lois Mulligan's lab at Queen's University to study the role of a particular protein in breast cancer. So very much a basic science related project, but there was lots of exposure during that time to epidemiology as well as clinical trials. Following that, I joined the Biotherapeutics Manufacturing Center, specifically the virus manufacturing facility arm of the group that's affiliated with the Ottawa Hospital Research Institute as a postdoc with Dr. John Bell to work on a BioCanRx funded project to build up capacity for accessible CAR T treatment in Canada. And specifically our group at the BMC, Biotherapeutics Manufacturing Center, so BMC for short, was focused on developing a clinical grade lentivirus production process, which we were able to successfully do, and then use that production process in a GMP environment to generate sufficient material for an ongoing CAR T clinical trial in Canada. More recently have moved into the role of head of R&D for the Biotherapeutics Manufacturing Center, and again, specifically the virus manufacturing facility arm, and really looking forward to the discussion today and happy to answer any questions that you might have.
Thank you, Piriya. Parvez.
Thank you, Megan, and thank you, BioCanRx, for inviting me. So my name is Megan mentioned, Parvez Vora. I'm the site head at Century Therapeutics Canada, and I lead this team of scientists here at McMaster Innovation Park, where we are developing this next generation off-the-shelf cell therapy for brain tumors. As for my background, I did my undergrad in India, where the major focus was zoology, but I was kind of exposed with a wide range of subjects including environmental sciences, molecular biology, immunology. And one particular subject that really fascinated me from early on was neuroscience. So I moved to the UK, where I did my master's in University of London, where my project was really focused on the cellular signaling by glial cell data neurotropic factor and its effect in the brain. Following that, I pursued a PhD at University of Manitoba in the cellular neuroscience group, where we discovered the various myelin-related genes in neurodevelopmental disorders. And really continuing from my master's research, I studied the molecular regulation of glial cells to form this normal myelin in the mouse brain. And as I was finishing my PhD, one thing I realized that I want to see my work reach the clinic, and that's what made me think that academia may not be the way to go. So I started thinking about what are the other directions I can go in. I knew neurodevelopmental diseases are too complicated to be cured, and we still require decades of research to really have a potent therapy. So taking my work experience in working with glial cells, I started researching different labs who are doing more of translational research. And that is where I stumbled upon Dr. Sheila Singh's research. So I joined Dr. Sheila Singh's lab at Stem Cell and Cancer Research Institute many years ago. It's almost 10 years ago as a postdoctoral fellow. And for those who don't know Sheila, she is a surgeon scientist and a world-leading expert in brain tumors. And my first project was to conduct some preclinical validation of small molecule inhibitors on this patient-derived brain tumor samples. Within few years, I was kind of upgraded. I became a program scientist, where I was spending more time leading the glioblastoma team. My role was to develop and design different efficacy experiments, not just with understanding the functional efficacy of therapeutic modalities, but also like monoclonal antibodies and cell therapies, along with chemotherapy, radiation, and things like that. So with that, I learned a lot about, what are the different options that are present for brain tumor patients. And we did a bunch of collaborations as well with in form of the SRAs, the sponsored research agreements with biotech companies, which exposed me more towards the biotech world as well. In parallel, I also got a MITEX Elevate Fellowship with Arch Biopartners. It's a small biotech company based in Toronto, where I manage companies. One of the portfolio products, Metamax, which are just set of peptides which can be used for imaging, diagnosis, or developing targeted therapies. The one thing which probably is not, I failed to highlight in my CV, I should, is I did a mini MBA with our McMaster GSA. They're running this, and maybe this is available in Toronto as well, but that kind of gave me a very, very brief introduction to MBA and what are the different things you need to know when you do this management studies. The reason why I'm telling you all this story is because I wanted to share all the detailed experiences which made me come where I am right now. And we realized that brain tumor is not an easy thing to go after. So I started discussing with Sheila an idea to kind of start a biotech company. So we spun out Piriya Therapeutics. Sheila was the face of the company. We had Jason Moffat, who was a gene editing expert based in Toronto. I, and Sheila, we started Piriya in 2018. I started as a director for preclinical development, but as with all startups, I ended up being the BD guy, the HR guy, the operational guy, and it was fun. There's strong evidence from science supporting the target that we were going after. There's a lot of data around cancer stem cells and conventional therapies failing in brain tumors. So there was a clear need for something what we are proposing. We published a paper last year in Cell Stem Cell, which showed the significance of using this CAR T therapy for brain tumor patients, and all that publicity ended up basically giving spotlight to Piriya. So Century Therapeutics, which is a US-based company, approached us a couple of years ago, and they were interested in collaboration, and we started talking about it, and they thought, wow, this is something that we would love to incorporate in Century. So we ended up getting acquired. But unlike usual acquisition, this was really opposite to that, where they would give us all the resources and all the expertise, where we would use our brain tumor models and use their iPSC-derived cell therapy platform and make a therapy for brain tumors. So right now, I lead the Century Therapy Canada site here. where the goal is to keep collaborating with the research teams in the US and building a strategy to treat these GBM patients, starting with a product that we can take into the clinic in a couple of years.
Thank you, and thanks. That was a great summary from all of you. And I think some of you spoke to it a little bit, but I guess the next question would be, what does your day-to-day look like? And Laura, I realize you're in academia. I think we probably know what your day-to-day looks like the most, but you had mentioned challenges, and I know you're an early career researcher, so perhaps you can highlight what your day-to-day might look like now versus what you anticipate it look like, in a couple of years from now. And so maybe I'll turn to you again, Laura, and we can use the same order for this next question as well.
Sure. Okay. So, I guess starting up a lab is a huge undertaking, and starting up a lab during a pandemic is a little bit extra. And so, I guess it depends on the time of year, but I've written a couple funding applications, and so that's taken up a huge chunk of time. I have a staff scientist who works for me, so we meet generally on a daily basis, and we have sort of a weekly lab meeting. We're trying to get cell banks, virus banks, all up and running, plasmid banks. There's a lot of like institutional protocols with which we need to be familiar in order to use the Animal Resource Center and to use, all of the capabilities that exist within BC Cancer. So, there's a lot of reading, and there's a lot of sort of administrative things that I do as well. But I'm really looking forward to, I have a PhD student starting in September, and so I'm really looking forward to when we're doing a little bit more science and we have a little bit more data to analyze, and that's the part that makes me excited, and why I got into it. So a lot of the administrative things are important, but not the super exciting part.
It's not why you did your training, is to do the administrative side. Well, congrats. That's really exciting. Piriya, what does your day-to-day look like?
And so each day is slightly unique. Sort of on a broad level, our main activities are working with our various partners, whether they be academic or industry, in terms of developing scientific processes, so both on the process development as well as the assay development side. And so lots of study design, writing study protocols, and in collaboration with our various partners for the projects that we're working on. A lot of once studies are being performed, if there's any questions that come up, there's lots of interactive discussions, both internally within the team, as well as communicating with our various partners. And then once the study is complete, then there's lots of data analysis and report writing again. And since we are essentially a contract development and manufacturing organization, most of this work is done in partnership and collaboration with our various academic and industry partners. And so in addition to that, another area specifically with regards to the R&D focus is that we also do innovative research as well too. And so we can further optimize and develop our overall processes to be able to increase the amount of yield we're able to get and offer to our partners down the road. And so essentially it's sort of two broad aspects that we're focused on, so that working the day-to-day with our various partners on studies and helping them essentially generate a process that can be used in a GMP to support early phase clinical trials. And then the other part of it is internal development to be able to expand our capabilities for our partners down the road.
Thank you. And Piriya, maybe just to back up a second, because some of our viewers may not know what a CDMO is, and I think you articulated it very, very well. But for someone coming in, what does it mean to GMP manufacturing? If you could just maybe speak to that for a quick moment.
Sure.
And the process development and R&D that obviously ties into it.
Yeah, that's a very good point. So essentially, if a group, whether it be an academic-driven trial or whether it's an industry, a biotech or pharma, if you want to generate material that can be used in a clinical trial setting, you have to be able to generate that material according to good manufacturing practices, so GMP production. And so what that entails is that various regulatory agencies, so in Canada, there's Health Canada, and the US FDA, there are certain guidelines that have to be met for material. And on a very high level, essentially, it has to be safe, like the quality has to be appropriate to go into patients. And so essentially in a GMP environment, people are gowned up, fully gowned up, and the environment is highly sterile and monitored, and there has to be no microbes or other things floating around there. It has to be super highly sterile. And so as part of process development in particular, usually various partners that we're working with might have a process to make their material of interest in a basic science lab setting. But one of the considerations to be able to support a clinical trial is you have to have lots of material. And so one of the areas that we focus in on when it comes to process development is to be able to generate lots and lots of material. And so that's upstream process development is essentially being able to generate sufficient amounts. And then other part, so our ultimate goal is to be able to generate material that's a quality that's appropriate to go into patients, comes into play with downstream process development. So the whole idea of downstream process development is to be able to purify the material, but there's a risk in essentially purification of material is that you might lose some of it. So it's a fine art, but it's essentially a lot of optimization and studies to be able to get the overall yield that you need, and still, most importantly, has to be at a quality that's appropriate for clinical trials. And I guess just very briefly, talked a little bit about sort of the departments that fall under R&D, but there's many other different departments within the biomanufacturing space that are absolutely essential in producing GMP materials. So for example, you have a facilities department, and the facilities department ensures that all the equipment in a GMP environment is calibrated and is behaving as it should. There's a quality assurance department that ensures that all the quality considerations in terms of all the regulations and all the processes are being followed. There's also a quality control team that ensures that the products are essentially the products that are required for the trials. So what I mean by that is you have to confirm the identity, for example, of the product, and so everything has to be tested. And then there's also lots of involvement with project management, operations, business development. So there's all sorts of different departments, and we'd be happy to answer more questions about any of that.
I'm on mute one time, every time. Parvez, and would you mind repronouncing your name? I think I'm mispronouncing it.
Parvez is fine.
Parvez, okay. Thank you. Go ahead. And so you remember the question. Perfect. Sorry about that.
So I do plan prioritizing different, ongoing research and operational activities in the company weekly, but also kind of, things start to kind of shuffle as you learn about different things happening in the company. Most of my time really goes towards the research effort, really project planning and tracking what is happening with the existing programs. A lot of time basically go towards coordinating the cross-site research teams. Also, I do most of the overhead work with the individual team members, empowering them to kind of, run or plan, key studies using in vitro and in vivo models that we have developed in the team. I do more of the writing the reports, attending lab meetings and the strategic scoping of what is a good project, how we want to kind of move ahead as a team in the company. A small percentage of my time basically go towards the operational part of it, where I'm also managing the facility and the equipments and really optimizing the scientific support that the group needs in order to kind of keep things running, and we don't run out of stuff, or we are not stuck with not enough resources needed to kind of move the program forward. So really overseeing that and everything is moving smoothly. We're sticking to the project budget. And also planning and executing other space requirements that we might have in the future. So again, a lot of things are done very quickly compared to academia and industry. The one thing which usually, there's a saying that, the industry doesn't have time, they have a lot of money. And while in academia, we have to kind of wait and see when the research grant comes by, and then we can start the experiment. So it does work the other way around, but so prioritizing is a key thing. There is always a number of things that you have to, but that's how I run the stuff right now.
Great. Yeah, thank you. And it sounds quite busy, not surprisingly, I suppose. So there's a question from the audience. And before I ask that, I just do remind others that they can post at any time. And I'll certainly defer to the questions from the audience first before I ask our moderate questions. The first question, though, is what would you have done differently, if anything, in your career path? What's the best advice you could give to a grad student or to your grad student self? And so, Parvez, maybe we can ask you first and go in the other order this time.
Sure. So in my mind, there's no rule book on how to be successful, and especially in the biotech world, because things are very fluid, things change. I always feel that you want to make sure that when an opportunity knocks on the door, you're ready for it. You want to keep learning and enhancing your knowledge constantly. You want to keep learning for that next big role, which might come to you anytime. So you want to keep developing the skills needed, and you definitely want to kind of proactively look for different opportunities. I don't think that I would. I have anything in my mind that I would have done differently. I think things like this it's an evolution process, right? Like, you meet people, you talk to people, and you learn more stuff. And then there is always the market, right? In the biotech world, whether the funds are available, and then who you're teaming with. There's a lot of team effort. There is never like one smart dude who can kind of start up everything. You always need a good team. So look for like-minded people who you like to work with to be successful. That would be a key thing in my mind. And I think there's always a luck factor, which I feel is super important as well, that you want to be there at the right time to kind of grab that opportunity.
Thank you. Go ahead, Piriya.
I agree with what Parvez said. I think there's many different paths to get somewhere, and it's all about sort of understanding what makes the most sense for you, given your interests and the opportunities that you're able to explore. And I guess one piece of advice that I wish I had earlier on is that exposure to all sorts of fields that are out there. I think early on I was very naive in understanding that you could go through academia or you could go through industry as possible routes after graduate studies. And so it wasn't until applying for various opportunities following a PhD did I come to have a fuller appreciation of the sorts of opportunities out there. So I would certainly highly encourage people to, as early as possible, to start talking to people, if you're in a lab setting, your fellow lab mates, your supervisor, to understand, what sort of opportunities are available to people. And definitely participating in BioCanRx HQP related events would be great for that as well.
Yeah, I echo that, Piriya. I think that's a great piece of advice. And if I may, I'd like to remind everyone that our virtual Summit for Cancer Immunotherapy is coming up. It's taking place on November 21st and 22nd, 2021. Registration to attend is free, and there are going to be some virtual networking sessions. So I realize that networking and meeting others is quite different in this virtual world, but there is going to be an opportunity for that. And again, just a shout out to BioCanRx. You can always follow us on LinkedIn. There's an HQP association group that has more information. So if anyone's interested in that, I'd certainly welcome any questions that you have. But thank you, Piriya. That's great advice. And Laura, to you.
I guess I would echo the other panelists in saying that the best advice is to be as much of a sponge as you can, to absorb as much from other people's experiences and to learn from that and integrate it into your own vision and your own sort of your own zeitgeist. At the same time, though, I would encourage everyone to challenge themselves and to make sure that the path that you're generating is your own. I moved to the States to do my postdoc, and I think, you asked whether there were things that we would have done differently, but there isn't anything that I would have done differently. I feel extremely lucky in having the opportunities that I have had, and I think moving to another country, to another, what is the States? It wasn't that far, but moving there on my own was, a huge challenge and a huge confidence boost, and it allowed me to, see things in a new way. And I think often when we stay in a certain place or exposed to a certain number of people, we can really internalize what all those other people want. And so it's really important to, have the space to decide what you want for yourself. And yeah, so the other just to come back to the luck thing, I do feel extremely grateful to my mentors who helped me along the way, because I think that those types of relationships are really what allowed me to be here today. And those are friendships that continue your whole career. So make sure to nurture those and express gratitude to those who have helped you get to where you are.
And Laura, that actually segues nicely into something that I just like your feedback and comment on. So me personally, I found that mentorship and sponsorship has been instrumental in helping me develop my professional skills. And by that, for mentors, I mean, very early in my career, I had reached out and talked to many different people, many different sectors, many different, levels of their career development. And in terms of sponsorship, it really truly developed into relationships with people who went up to bat for you. And so I'm wondering if any of you guys have your own mentors. It sounds like, Laura, you do, that have become friends and also sponsors. And if you do, what advice might you have for our HQP in terms of developing those types of relationships?
I can take that. I think I would definitely name Sheila as one of the key mentors I had in my career success, or if I won't say it's a success. So I think one of the things people should remember is everybody comes with skill sets that you can take stuff from, right? Sheila had a lot of key advice for me in terms of my career building, in terms of science and how to take the science forward, and problem solving skills and things like that. I learned from her. There were other mentors for me in terms of management. When we start talking about biotech industry, unfortunately, most scientists are not good in managing. And I'm not saying Sheila is amazing, because she has another role as a neurosurgeon as well. So she does manage a lot of people, but most scientists are not good in managing. So what I did is I found a mentor who would teach me skills on how to manage a company, learn skill sets which are required to raise money or do drug development work. And those are the skills you have to get from somebody else. So my advice would be look for multiple mentors. Like one person may not be sufficient for your career success, and very likely you will find as you move up in your career ladder, there will be different kind of requirements you'll have for your success. So look for the right person as you move up. But coming back to what Laura mentioned, and I should mention that as well, that be grateful to the people who have helped you and pushed you forward. So definitely keep those relationships. Like I've seen some people, burning those bridges after they had. That's it always comes back. And again, Canada is a very small community, so make sure that you cherish those moments that you had with your mentors, and keep in good relation terms with them. And you never know, maybe you end up starting a company with them down the lane.
Perfect. And do either of you have anything to add?
Yeah, so I guess I would also say that, my PhD supervisor was a mentor in so many ways. John really changed my life and inspired this like really passion and creativity and excitement about science, about translational work, about, working towards developing something that's going to make a clinical impact. And so that really launched my career, and that's, I think, such a core part of my mindset and the way that I conduct myself at work and the way that I motivate myself to keep going when experiments don't work. And then I did a postdoc, and Richard was fantastically creative as well, and just some of the conversations that we have about where we can take things, I think that's really, really exciting. And then the other person who was a really core aspect of my mentorship team, I guess, was a collaborator from Duke. And he was the one who actually ended up giving us the CAR construct, which we worked on at Mayo. And he really helped us, initially just to troubleshoot things moving forward, but he became such a close friend, and he really helped to, shape the research program that I'm developing at BC Cancer. It's really not obvious. Nobody tells you how to interview for a PI position. And so, there's a lot of not obvious things that you need to be able to present in your application package. Anyway, so he was extremely, extremely helpful. And I'm extremely grateful to him as well. So, and now I have a new mentorship team here at BC Cancer. And so I'm really excited to start to build some of those new relationships.
Thanks. And maybe, actually, Piriya, sorry, did you have anything you wanted to add?
Very quickly agree with both Laura and Parvez. I think each of the mentors I had throughout, from undergrad all the way to the postdoc and beyond currently as well too, have each been instrumental in helping me in my career aspirations and journey. And so definitely would encourage people to nurture those relationships for sure.
Thanks. And then I wanted to pick up on something you kind of all highlighted, and that's that you had really, really excellent mentorship in your PhD or postdoctoral supervisors. And so one of the questions that often comes from our undergraduate student population especially is, how do you pick a supervisor? How do you go about picking a research program or project when it comes to being in an undergraduate where you could pick anything? So maybe if you have a few words of advice on that, is a big question, but what are your thoughts about that?
I can go first for that one. So when I completed my PhD, I was definitely, you want to look for a supervisor who is doing the research which you are interested in. And then I did it. did it, I think, too systematically. I made a list of people, I prioritized them, that this is the PI I want to really go with, and things like that. And really, when you were doing that, you want to start with the best of the best, right? And after I made the list, I look at the profile of those people, and I'm like, there's no way Sheila is going to take me. Or, there's another PI who is an amazing neuroscientist at University of Calgary, and I'm like, okay, this guy is like top level. Sheila is top level. They're not going to take it. And it really comes back to our first question was how mentors help you. So one of my PhD committee members, I went and talked to him. I'm like, I like this guy, but I don't know if they'll take me. Like, I'm just like, a guy from Manitoba, PhD is not like really the best one. These are like high profile pictures. So, and he said, if you don't apply, you will never know. Why don't you just make an application? I think you start with the top people that you want to work with, and then work your way down. And if you get rejected, don't take it personally. I have met some of these PIs who did not take me. And after I met them, like I came to know that they did not have grants, or they were just waiting for the money to come by, or they just recruited somebody else. So it wasn't really against me. But for me, I think my part was to do the application, which I did. Like I did start with that application early on, and I was fortunate that by the time Sheila was the second in the list, it took me some time to kind of go back and forth because I was in Winnipeg, had to come for the interview. So things were pretty different then, but I was lucky there for sure. But start with the top application. And I actually wrote an email back after I came here to my PhD advisor, who actually told me, is like, go for it. Like, if they don't respond, when they don't respond, you never will get it. That's fine. But if you don't apply, you will never know. So go for it. Like, I think you want to show your best to these people and see if you can catch a break. And sometimes they do come back after 2 or 3 months. Sometimes if the grant comes back, they're like, oh, this was an excellent PhD student who contacted me before, was really keen in learning more about X and X research that they wanted to do. So you will definitely get the place if you show that sincere effort in joining their group.
Great advice. I think classic expression is you miss all the shots that you don't take, right? Classic hockey expression. Great advice. I think that's well said. Laura or Piriya, do you guys have anything you wanted to add?
Sure. I guess I would encourage you also to start as early as possible. I was really lucky to work for John during the summer. And so I worked for him during the summer. I did an honors project. I started a master's project, and then I did my PhD. And so I just, I never left. But, having that initial early connection, I think, was really important. And even if that first one that you engage in isn't the thing that like really sparks something exciting, I think it's having that experience is super important. It looks great on your resume. But it helps you to generate a scaffold of what you're interested in and what perhaps you're not interested in. And then the other aspect is, maybe that person who you work for will then go on to send an email and say, hey, I know this great summer student, maybe you'd consider taking him or her on. They were great in my lab. And so I think coming back to the idea of mentors and building your network, those types of relationships are particularly important.
And Piriya, do you have anything?
I would echo what everyone else has already said, and add that I guess when you're trying to identify who a suitable supervisor might be, it might also be helpful to, when you're meeting with them, to also meet with their lab as well too, to see sort of the research interests, the dynamics in the lab, if they're going to be a good fit for you as well too.
Yeah, really, really great advice, and quite not unique, but very different perspectives from all of you, but obviously all fitting in really, really well together. So again, at the beginning of the session, I said I'd leave the last 15 minutes for us to stop recording. I think I'd like to change that if everyone's okay with it. We do have one more question coming in, but it's been relatively quiet on the Q&A chat box. I think that's successful for us, but at any rate, I'd like to keep it open because we often get a lot of viewership afterwards. So I think if you're okay with it, we'll just keep recording and post the entirety of the session. So one question that came in is, what's next? What are your future career aspirations? Yeah, so I'll open it up to all of you for this question.
I'll let Laura or Priya go this time, because I feel bad, like, then I take all the key points first, and then they have to say I agree with Parvez, and so let me give them a chance first.
Well, I think your career trajectory is going to be all of our career trajectories are going to be very different. So you may not be taking all the good stuff for your own. I think that I'm starting my lab, and so things are very new, but I think for the next little while things are going to look pretty similar. Obviously with a lot more students, a lot more staff. I'm really looking forward to really getting the projects going. Hopefully getting lots of successful grants so that we can bring in more people. But I do think that, it's been a year, it's been a year of a lot of change. And so I'm really looking forward to just sort of focusing on the science and sort of coasting in the scientific space a little bit.
I guess I'll add, I really enjoy the biomanufacturing space, and so do really want to continue in this field. I think we've all realized with the global pandemic and how important it is to have capacity to be able to produce treatments as well as vaccines, that the space is only going to grow within the world as well as within Canada as well too. And so I am very interested in continuing in this space. And I do think contract development manufacturing organizations in general do play a really interesting role in the overall ecosystem of Canadian or just broadly scientific innovation. And so I'm quite excited to see how the field continues going forward.
I think one of the things I've been working on since last 1 decade is brain tumors, and I personally would really like to see if I can, play a major role in taking a drug all the way to the clinic. So that has been my goal since past 3 years since Empirica, but now I think it's a real possibility with working with Century. I think the goal is in the coming years, if we can take this to the clinic and see some big effect. The conventional therapy for brain tumors haven't changed in the past 3 decades, especially for GBM, and seeing something change in the clinic would be a big win-win for our team, I think. So that has been in my mind since we started with Empirica and Century.
Yeah, that's great. Certainly a good aspiration, good aspirations from all of you. So thank you for that. We do have another question from our participants, and that is, what type of reading do you do, both in your field and outside of your field? And how do you manage your time? I think it's getting at that work-life balance as well. So Piriya, maybe turn to you. It's true, we are going in circles. So how about we go to our middle person answering first?
Sounds good. In terms of readings within the field, a lot of different process development related publications to sort of understand where we might be able to design some experiments to further optimize the processes that we're working on. we ever have a particular study that we're working on and need to do some further optimization, a lot of this will be coming from literature that's available in our respective various platforms that we're working on in terms of what others may have done. And in addition to reading the various papers and publications that are out there, find that there's a lot of webinars and also blog articles as well too on these topics, which are helpful. So for example, during a quick lunch, if there's a webinar that's particularly relevant, you're able to sort of balance time there. But I do think it's important, sort of related to the second question of how to manage and balance time. It's all about setting out the priorities in a particular week, and those, given the field, and I'm sure relatable to many different positions, is that priorities will slightly change. But essentially carving out time for your priority in work related activities, and then for your personal life as well, and closely monitoring that would be important, and my suggestion there.
Thank you.
So I also work in immuno-oncology, as I think Laura and Piriya both are, and the field is evolving very, very fast. So to keep up to date, definitely kind of do some reading and manage a time every week that this will be 1 hour or 2 hours of this particular day where I'll be spending just looking at what things have changed, what are the new publications coming in, and not just in the area of research that I'm in, but also other indications, because there's a lot of learnings that you can get in fields which are far more advanced in terms of making immuno-oncology therapies. Like melanoma is far ahead, and of course all the different blood cancers are far ahead. So there'll be a lot of learnings that we get from those preclinical as well as clinical trial data that we can use in the GBM or the brain tumor research. So that is one thing. The other thing is also having an opportunity to work with people who have actually done a lot of drug development. I think interactions with those people are also part of the learning that I'm fortunate to have. So that's where my second aspect of learning comes in. In terms of management, I think it's very difficult. The field we have chosen is evolving super fast. So there are days where you are super storm, you're working like straight 12 hours a day, and the days where things are slower, you're just waiting for cells to grow. So those are the slow days. So there is excuse me, there's never a clearly defined work-life balance. Unfortunately, but that's what we have chosen. It's not a bank job that you can go to 9 to 5 and come home and you're done for the day. But I think as long as you have the passion and the energy to keep doing what you're doing, it's great.
I guess I would really echo what Parvez said about reading in disciplines that are outside of what you really consider your core niche. I think that some of the best ideas really come from adjacent fields. And when I moved here, I wanted to establish some lymphoma models, and I was really heavily focused on solid tumors. And it's sort of like a solid tumor, but there's a whole field of things that I hadn't appreciated. And so I've learned a ton by trying to immerse myself in a brand new field with new techniques and a completely different vantage point. So I would encourage you to expand your horizons as much as possible. But in terms of prioritizing what papers to read, I joined Twitter when I started my lab to try and generate my presence. And I really only follow a handful of other scientists, but if you follow some people who are relatively active on Twitter, that's where everybody posts their papers, and I think that's a really nice way to stay current on what's being published. And yeah, especially, if you can follow some people who are in your field or in your institute, or gives you a good sense of what's going on. In terms of having a work-life balance, I don't know. There's no good answer. I think the answer is you just do the best you can with what you have, and if you're passionate about your work, then I think that'll come through. And it's important to manage your relationships at home, because that's really what gives you the sustenance to keep going.
Well said. So we're rounding the end of the hour. We have about 4 minutes left. So just before we have closing remarks, I did just want to say that a pop-up survey will come up at the end when you close down this window and end your session. And we do ask that you fill that out, because it provides a lot of feedback to us in terms of developing these types of webinars and training opportunities. So just a note on that. But maybe just to close us out, a final kind of open-ended question, and that is, what advice do you have, or is there anything that I didn't ask that you think is burning that the audience might really want to know about? We did well. We were comprehensive, in terms of asking these questions. Maybe in that case, final remarks.
I can add one of the things which many of you might face is when you start looking for jobs, whether it's in academia or industry. I think, and I did allude to it a little bit, but maybe I would love to kind of stress a little more, because many people get really discouraged when you start making these applications and nobody replies. Unfortunately, because there's so many applicants and so little number of jobs available, we don't get responses every time. So don't be discouraged. I think you want to just keep applying. Make sure you are tailoring your job applications very well, and do use social platforms, as like Laura was mentioning. Twitter is a great place. LinkedIn is something which I wasn't like a big fan of early on, but in the last few years, I set up that account, because you learn a lot. Even I think all social media platforms are, if you're using it in the right form, I think that could help you. So be sure to kind of, reach out to people, talk to them. I have emails from people who's like, oh, can we go for a coffee? And of course, I cannot go for a coffee like with strangers, but then the extent to which people are being aggressive in getting their jobs makes me feel that, sometimes the deserving candidates might lose an opportunity if you are not really assertive about what you're looking for, and if you don't come in contact with the right person at the right time. So don't hesitate, that, oh, what will somebody think? Reach out to them, LinkedIn, email, phone, and talk to them about, how they reach the place they reach, and what job opportunities are there in their field, and if there's an opportunity in their company. So that's my final advice.
Thank you.
Maybe I'll just frame my final advice in the question in the Q&A, which is what personal attributes are essential. And so I guess there's 3 things that I sort of want to emphasize. One is to be an expert in whatever you want to delve into. 2, to be really passionate about it and to be really excited about it. And that's what forges connections with other people, and that's what keeps you going. And then the third thing is that perseverance and grit are what get you through. I'm certainly not the smartest. I'm not the most creative. There's lots of other people who are equally as deserving to be in this position to me. And there's a lot of luck involved. So just keep going until you get your shot at it.
Just quickly, I guess, in terms of what personal attributes are essential for success, and we'll use it as my final advice as well too, is having a resilient mindset is super important. There's many times during whether your graduate school, during your career, or in life in general, that things might not go as expected. So if you're able to adapt, be positive, stay optimistic, and sort of take the best out of the situation, then it'll always be very, very helpful and key in a professional and personal setting.
Excellent. And sorry, Parvez, did you unmute to say, did you want to add something? No. Okay. Well, we're at 4. That was really, really excellent. Thank you all for your advice and your feedback. Really appreciate your time and your commitment. And it was really cool to hear your different career paths and where you are now and where you think you'll be. So perhaps we'll catch up with you in a couple of years from now again. Until then, this has been recorded. We will be, posting it on our social media and LinkedIn and promoting it. So so stay tuned for that. But otherwise, have a good day to everyone, and we'll see you again soon. Thank you.
Bye. Okay.
Bye, everyone.